All right, so our next speaker is Raphael Loll-Krippleber. I hope I'm pronouncing that right. From the Brown Lab at University of Toronto, who's going to talk about P-body regulation.
Hi, everyone. So I'd like to start by thanking the organizer for giving me the opportunity to present my work today. And as Audrey said, I'm going to talk to you about P-body regulation of mRNA abundance during DNA replication stress. So P-bodies are membrane-free granules that forms in the cytoplasm of eukaryotic cells, and this formation occurs after different kind of stresses, such as glucose starvation or osmotic shock. And P-bodies are structures that are important for mRNA degradation and mRNA storage. And so here I'm showing you an example for three P-body components that form granules in response to glucose starvation, DHH1, which is an RNA helicase, PAT1, which is an RNA decapping enzyme activator, and LSM1, which is also involved in RNA decapping. Can you go on the next slide, please? So a couple years ago, we found that P-bodies are also forming during DNA replication stress induced by treatment with hydroxyurea, which is a DNA replication inhibitor. And the Moreno's lab also found that actually LSM1 is involved in degrading histones mRNA and during replication stress. And this is illustrated here on this northern blot, where you can see that the LSM1 mutant is not degrading HHG1 mRNA upon HU treatment, while the wild type is clearly degrading it. And so this response, the importance of this response is illustrated by the fact that LSM1 delta strains are actually sensitive to hydroxyurea, but if you delete histone genes in an LSM1 background, you are suppressing partially this sensitivity. Can you go on the next? So we hypothesized that P-bodies might degrade other mRNAs to prevent the production or the accumulation of proteins that might be toxic for the cells during replication stress. And so we decided to perform a screen to identify these mRNAs, and we can use the same kind of approaches than the Moreno's lab, which I'm showing you in the next slide, please. So we used two different screens. One screen was to measure mRNA abundance in P-body mutants during replication stress, and this was with the aim to identify mRNA that are stabilized during replication stress in absence of a functional P-body pathway. And the other screen was a suppressor screen, and so this was aiming at identifying genes with inactivation suppresses replication stress sensitivity of P-body mutants. Can you go on the next? So we performed RNA-seq experiment on LSM1 delta cells before and after 1, 2, and 4 hours of HU treatment. And as expected, we found genes to be more abundant in the LSM1 delta mutant. And so this was the proportion of upregulated genes increased drastically after HU treatment. We were also, and so these genes are shown here in yellow on the heat map, and we were also surprised to find genes to be downregulated in LSM1 delta. So this was already described in other degradation, mRNA degradation mutant, and I'm going to come back to this in a minute. So using our RNA-seq data, we were able to identify 829 transcripts with abundance increase in LSM1 delta during at least 2 time points in HU. And among these transcript we identify 11 candidates that were also suppressor of LSM1 and PAT1 HU sensitivity. And we were able to validate 6 of them. So these 6 targets were revalidated using a spot assay, and I'm showing you here the spot assay for these 6 targets. And as you can see, deleting these 6 targets are actually suppressing LSM1 and PAT1 HU sensitivity. And we were quite pleased to find HHT1 among these targets. As I told you, HHT1 is an already known Peabody targets during HU. And so today I'm going to focus on YOX1 for a few reasons. Can you go on the next? So YOX1, we identify YOX1 in another suppressor screen that we did in collaboration with Corinne Islow's lab at UBC in a liquid culture suppression screen. YOX1 is also involved in the regulation of DNA replications and cell cycle regulation genes. Overexpression of YOX1 induces chromosome instability, and YOX1 relocalizes from the nucleus to the cytoplasm upon HU exposure. And so the way YOX1 is thought to work is that it can bind to MCM1. And so the binding to MCM1 actually repress MCM1 transcription of MCM1 targets. So YOX1 is a transcriptional repressor, and I'm just going to ask you to remember that for the rest of my talk. Sorry. So the first thing that we wanted to check was whether we could detect YOX1 transcript at p-bodies. So for that we used the MTAC system, which basically use mRNA binding protein fused to GFP, and this mRNA binding protein can bind to repeats that we integrated in the 3' UTR of YOX1. So we first visualized YOX1 mRNA in wild type, but we couldn't detect any YOX1 granules, and so we reasoned that maybe YOX1 was being degraded in the wild type at p-bodies. So we deleted XRN1, which is the major mRNA exonuclease that localizes at p-bodies, and in this case, in the XRN1 mutant, we were able to detect YOX1 granule formation. Can you go to the next? So we co-express our MTAC system in a strain expressing a p-body marker, PAT1 mCherry, and we were able to actually co-localize YOX1 transcript with PAT1, both in untreated condition and after HU. And so after HU was a little bit more difficult because we were detecting large PAT1 aggregates, but when considering discrete foci, we were able to see a colocalization in 25% of the case in both untreated and treated condition. Can you go to the next? So the next question was, so I showed you that YOX1 mRNA transcript is more abundant in LSM1 delta cells, so we wanted to see whether the YOX1 protein was also more abundant in LSM1 delta. And so we measure YOX1 GFP intensity in the nucleus of the cells, because YOX1 is a transcription factor, and we were able to detect actually that YOX1 protein is more abundant when the p-body pathway and LSM1 and in LSM1 delta cells when LSM1 delta is when LSM1 is inactivated. Can you go to the next? Okay, so far I showed you that the YOX1 transcript is increased in LSM1 delta cells. YOX1 protein is more abundant in the nucleus, and YOX1 is a transcriptional factor, so transcriptional repressor. So the next question that we wanted to ask was, is an excess of YOX1 protein results in the downregulation of genes that are important for HU resistance? So again, can you go to the next? Again, we use the RNA-seq approach, and this time in cells overexpressing YOX1 and before and after HU treatment. And as you can see on this heat map, a large proportion of the genes are downregulated when we overexpress YOX1. So these genes are indicated in blue. And so we wanted to see whether these genes were actually also induced by HU treatment. So can you go to the next? And this is the case, as you can see that a good proportion of the genes are actually in yellow in the HU heat map. So that means that these genes are induced by HU and they are downregulated by YOX1. And so can you go to next? And so we also wanted to see whether these identified YOX1 targets were also downregulated in LSM1 delta, and this was the case for 1/3 to 1/2 of the YOX1 targets. Can you go to next? So we wanted to see whether we could actually identify targets of YOX1 that are actually important for HU resistance. So we reasoned that these targets should be downregulated in LSM1, and also that inactivation of these targets in an LSM1 background should render the cells more sensitive to HU. And so using these criteria, we were able to narrow down to a list of 8 genes to test. And so the way we tested them was to actually overexpress these genes in an LSM1 delta background and see if overexpression could suppress HU sensitivity. Can you go to the next? So what I'm showing you here is the data for 2 genes that we tested and that were positive. So I'm showing you the growth curve of an LSM1 delta strain with an empty vector in black, and LSM1 delta strain overexpressing either ICS2 or ALD6. And you can see that when you overexpress these 2 genes, you grow faster in HU. So these 2 genes are actually suppressing LSM1 delta HU sensitivity. So can you go to the next? In summary, today I showed you that YOX1 mRNA abundance is regulated by P-bodies / LSM1 in a manner during replication stress. I showed you that the P-body dependent regulation of YOX1 abundance prevents the accumulation of the YOX1 repressor in the nucleus, and this response is important in order to prevent the repression of genes that are important for HU resistance during replication stress, and these 2 genes are ILD6 and ICS2. And so with that, can you go to the next? I'd like to thank the members of the Brown Lab, and especially Grant Brown, Dax Doherty at the Donnelly Sequencing Center for his help with RNA-seq, and Grant Tran in the Nislos Lab for his help with the suppressor screen in liquid culture. And I'll take a question, and I thank you for your attention.
I have a question. So just actually sort of a technical question and then a question of clarification. So technically the aptamer, the MS2 aptamer, we've looked at it in different transcripts, and it can really affect the stability or the translational efficiency in 3' UTRs of different transcripts. So I'm wondering if you've got orthogonal ways of testing that in this case to make sure that the MS2 aptamers aren't messing things up.
No, we haven't tested whether translation is affected.
Yeah,
but it's possible that there is a level, because if you look at the protein levels, it doesn't mirror the YOX1 mRNA levels, for example. So there must be another layer of regulation in this case. The
other question is, so in the XRN1 plus mutants, you have like no localization to P-bodies of this aptamer tagged transcript, right? And
we do see a little bit, okay. But it's so
just
rare.
A question of very reduced abundance of the transcript, right?
Sorry, say that again?
Is the transcript abundance really a lot lower when XRN1 is there, or?
We haven't really looked into that. Yeah, I can't tell you whether XR1 deletion actually like really increases. It must be increasing the transcript, but I can't tell you like what the extent of the stabilization in that case. Right. For the LSM1 mutant, for example, it's between 2 and 3-fold increase.
Sure.
I don't know in the XR1 delta case.
Yeah, in the case though, I mean, even upon replication stress, you're not seeing this go to in XR1 plus mutants, you're not seeing it go to the puncta. And so I'm trying to figure out where is the regulation if it's already so unstable. Anyways,
but we don't know. It's a good question, yeah.